this is the granddaddy of them all, the action potential, the nerve impulse. So in my mind, this is the hardest part of Psych 2. This is one where students really seem to have a difficult time. So what I'm going to do is I'm going to go through the basics of it, and then I'm backed it up with different videos. So hopefully you'll hear the basics, you'll have the book, and then you'll see the videos, and you'll be able to get through it. So some of you are saying, Amy, if it's so hard, then why do we have to learn it? Because the action potential and the nerve impulse, it's the root of life. There is no life without an action potential. Every thought that you think is an action potential. Every movement that you do is an action potential. Every emotion that you feel, every sensation that you experience is all just action potentials. So basically what we're looking at is we're looking at what is life. We're looking at the action potentials. So what I'm going to do right now is I'm going to show you a little video to give you a broad overview of what we're going to be going over, and then I'm going to take you through it step by step.
All neurons or nerve cells consist of a cell body, an axon, and one or more dendrites. Dendrites receive signals from other cells and carry them to the cell body. The axon carries signals away from the cell body and toward other cells. The nervous system carries signals through the body in the form of nerve impulses. A nerve impulse is a wave of chemical and electrical change that moves along the membrane of the nerve cell. Between impulses, the membrane is said to be in its resting state. In the resting state, the inner surface of the nerve cell membrane has a more negative charge, and the outer surface has a more positive charge. This difference in electrical charge across the membrane is called the resting potential. When the nerve is stimulated, channels in that area of the membrane open, and sodium ions, Na+, flow into the cell. The flow of these positively charged ions reverses the charge difference across the membrane. The inner surface becomes positive and the outer surface becomes negative. The reversal of charge in this region of the membrane causes channels in the neighboring region to open. Sodium ions flow in and the charge is now reversed in this region. As gates open in one region after another, the nerve impulse moves along the membrane. A few thousandths of a second after the sodium ions enter the cell, other membrane channels open and allow potassium ions, K+, to flow out of the cell. This outflow of potassium ions restores the membrane to its resting potential. This reversal of charge, followed by restoration of the resting state, is called an action potential. Action potential is another name for nerve impulse. Action potentials travel very rapidly along the membrane of a nerve cell. Some move at a speed of 300 feet per second.
Okay, so that video showed you the basics of what we're trying to learn. I know it kind of went fast. Now what we're going to do is we're going to break it down really simple, step by step, so we can get it in our notes, okay? So let's start out with this term, resting potential. So we're starting out with a neuron at rest. So let's go ahead and put these on. So we learned that this is a neuron. Okay, so I'm drawing the axon. And I'm drawing the cell body, and now I'm putting the dendrites on. Okay, let's put the nucleus here. So when this neuron is at rest, this outside portion of the membranes here, the outside of all this that I drew, it's like a sponge. It's a semi-permeable membrane. Oh, one more thing is resting potential, like in this particular chapter, there's a lot of synonyms that mean words that mean the same thing. So when I say the neuron is polarized or the neuron is in a state of polarization, it means it's at rest. Resting potential means polarization. So right now the neuron's not doing anything. The outside of the cell is like a sponge. It's a semi-permeable membrane. So here's a picture here. See, this is the outside wall. So here, do you see how these small ions, molecules can go in and out of the sponge? This here, these bigger ones can't go in and out, so that's why it's semi-permeable. Some things can go in, some things can go out. Okay, so this is that. So now we're going to go to this next slide and start to understand some more. Okay, so you saw those little pictures on the other side, these things right here. What these would be called are ions, and ions are charged particles. So in your textbook, they're going to go into sodium, potassium, and chloride. However, it's been my experience that just learning the sodium and potassium is enough. The chloride you can add at a later level if you go on in the biopsych and neuroscience. Then every single class that you take is going to be going over this action potential thing. They're going to go over it faster than me. They're going to go over it probably in a little less depth because this is the introductory. So what I'm trying to do is give you your first layer. So we're just going to work with sodium and potassium, and then you can add the chloride like at another level. But this one, I just want you to get the understanding of sodium and potassium first. Okay, so notice that both of these have a positive charge. Okay, so really pay attention to that. So I'm going to draw this again. Okay, so dendrites, axon. Okay, here we go. All right, so here. When the neuron is at rest in a state of polarization, it actually carries a charge of -70. And you say, wait a second, Amy, why would it carry a charge of -70 when it's at rest? Because on the inside of the cell, there's all these different protein molecules that carry a negative charge. So how we know what it is like an oscilloscope, and here it's testing, it goes in to see what's in there, okay, right? So -70 millivolts, but we're dealing with these ions, and what this action potential is going to be is the action potential is going to be the movement of these ions, basically. So when the neuron is at rest, the K is trapped on the inside, okay? And you say, but K is positive, but you said it's negative, okay? Well, if K wasn't in there, it would be more negative. It would be more like -120 or -100. So the K does have its positive charge in there, and it's balancing out the negativity of the protein ions that are trapped inside. That's all I can tell you is when the neuron is at rest, there's more K on the inside, okay? But then Na, sodium, is on the kind of more masked out on the outside. So look, the outside of the neuron is a semi-permeable membrane, and it's more permeable to potassium than it is to sodium. So these big red ones would be more like sodium. We could just probably write it right here. This would be more like the sodium. And then these little pink ones, you see how they can go in and out? It's more like potassium. Okay? So what I do in my face-to-face class is I have 2 cups, and you can see through them. They're like big glass cups. And then I have this white sponge. So in this cup right here, I mix in some sugar. I mix it all up in the water. And then this cup right here, I take some pepper and just mix it in the water. And then I have a white sponge. So if I dip the white sponge into here, would you be able to see the sugar on the outside? No. But you would know that is the sugar going in and out? Yes, some is being kept out, some is going through. So this would be, the sugar would be more like potassium, okay? But here with the pepper, it would be more like sodium, because when I mix up the pepper, you can still see it in the water. I stick the sponge in. When I pull the sponge out, you can see the pepper on the outside. But is there some pepper that's getting through? Yes, there is. But, you know, most of the Na is being kept out. So when we've been talking about a semi-permeable membrane, it means that the outside of the neuron is kind of like a sponge. It lets some things through, keeps other ones out, but things can slip through, okay? So let's just review what we have for resting potential, and I'm going to show you a video to reinforce this, okay? So when the neuron is at rest, it's said to be polarized. It's in a state of polarization. The outside of the cell has a semi-permeable membrane, which keeps things out and lets other things go through. When the neuron is at rest, there's more K on the inside, but the charge of the neuron is -70. Oh, your book goes into -30 to -90. Okay, that's true. So neurons have a different resting potential, but for the sake of this particular lecture, we need to like pick an arbitrary number, and it's very, very common for intro biopsych textbooks to use the number -70. Very common. But just for your own note that each neuron might have a different resting potential. Okay? So one other thing is a way that you can keep that which one is on the inside and which one's on the outside is I try to use this kind of thing. It's okay for potassium to be inside, but sodium is not allowed. Okay? Maybe you can try that. Alright, so let's go watch this video and see if it can be reinforced visually.
Unstimulated, neurons maintain a constant electrical difference or potential across their cell membranes. This potential, called resting potential, is always negative inside the cell and ranges from -40 to -90 millivolts. If a neuron is stimulated, the negative potential inside the neuron can be made either more or less negative depending on the stimulus. If potential is made sufficiently less negative, it reaches a level called threshold, and an action potential is triggered. During the action potential, the neuron suddenly becomes 20 to 50 millivolts positive inside. Action potentials last a few milliseconds before the cell restores its negative resting potential. The cell membrane of a neuron encloses cytoplasm with various ions dissolved in it. The neuron itself is immersed in a salt solution, the extracellular fluid. The ions of the cytoplasm consist mainly of positively charged potassium ions and large negatively charged organic molecules, such as proteins. Outside the cell, the extracellular fluid contains mostly positively charged sodium ions and negatively charged chloride ions. Since charged particles cannot pass through the lipids that make up cell membranes, they must travel through channel proteins that extend through the membrane. In an unstimulated neuron, only potassium ions can cross the membrane, traveling through specific proteins called potassium channels. Although sodium channels are also present, in unstimulated neurons, they remain closed. Since only potassium ions can cross the membrane, and potassium ions are more concentrated inside the cell, they diffuse out of the cell, leaving the large negatively charged organic ions behind. As more and more positively charged potassium ions leave, the inside of the cell becomes increasingly negative. But since opposite charges attract, as potassium ions diffuse out, an electrical force develops that tends to pull them back inside. At some point, the diffusion of potassium ions out of the neuron due to concentration differences is balanced by the electrical attraction tending to pull them back inside. This is the point at which neurons reach resting potential. Reaching resting potential in this way does not require significant changes in the potassium concentration inside or outside the cell. Only about 1/10,000th of the potassium ions initially inside a neuron must leave to create a resting potential of -60 millivolts. But it is action potentials, not resting potentials, that carry information through a nervous system.